So we're going to have 3 speakers, actually 2 live speakers, and then Augusta Rendon, who is going to give a live talk, is actually going to give a pre-recorded talk from the GIF meeting because he fell ill. So he won't be here for the Q&A session, but we're thinking of him. And so we're going to have 3 different talks, and I'll introduce each one as we go, and then we'll save questions for after the 3 talks at the end with using the Slido function. So our first speaker is Shaikh Farhan Rashid. He's a senior application development specialist at the University Health Network. He works on the CanDIG project, which is a distributed genomics platform, data platform that we'll hear about shortly. And he's been working on the application of the Global Alliance Cloud API standards to the CanDIG platform. So I'm going to turn it over to him to start his talk. Thanks, Shaikh.
All right. Thank you, Heidi. Hello, everyone. I hope you are in good health and in good spirits, or at least sufficiently caffeinated for my talk today, which will be on the Federation of Health Research Data in Canada. And my name again is Shaikh Farhan Rashid. I am a member of the CanDIG project based out of the University Health Network in Toronto. So the presentation will be as follows. We will start with a look at the history of healthcare in Canada and its impact on health research data. And then we'll look into federation, what that means, and as well as exploring a couple models of federation and ultimately CanDIG's federation model and how it helps to bring data together across across many provincial sites in the Canadian landscape. From there, we will go into the GA4GH and how it has helped CanDIG in reaching this goal of connecting Canada's health and genomics data. Then we'll get into the more technical part of this presentation, which will be looking at our CanDIG V1 to V2 transition, the re-architecting of our CanDIG platform, and where we have implemented GA4GH standards and APIs in order to make this in order to overcome some of these technical challenges with some specific examples. And then we will conclude with a look into ways that CanDIG can continue to contribute in the GA4GH in both technical and standards and policy making places where possible. So every day, genomic data is being produced at sites across Canada, but accessibility of that data to researchers continues to be a challenge at the national scale. In Canada, healthcare and therefore health research data is a matter of exclusive provincial jurisdiction. Each province has made its own decision about privacy trade-offs based on its population's own choices, and that has real-world impacts on the ability for researchers in Canada, healthcare researchers in Canada, to be able to access data outside of the jurisdiction of the province which they reside. One solution would be to think about centralization of data, but as we've just discussed, that bringing this data together in a national way would require many changes and considerations of provincial statutory and regulatory requirements, and that makes this process very difficult to do from the perspective of centralization. However, when it comes to federation and thinking about solving this problem as federation, it is a way that we would be able to resolve some of these political challenges and legal challenges while still being able to respect the legal framework that currently is in place for healthcare and health research data. And before I go any further, I do want to say that even though that my presentation today will be about health research data, the focus of it will be specifically in genomics data. So federation, what is it and what does it mean for us in CanDIG? So in the political sense, we see federation is a union of partially self-governed provinces, states, or other regions under federal government. And in Canada, that is very much the case, where our federal government is the main sovereign power, which has delegated through the legal jurisdiction of health care and health and the jurisdiction of health research data to the provinces. But in the computational world, the world that I'm more familiar with, I think of federation as a system of connected networked computers. But moreover, we can think of it as a set of network systems agreeing to share moreover, we can think of that of a federation as a network systems agreeing upon standards of operation in a collective fashion. While this model is interesting to us, it's very applicable because in the computational federation model that we will explore today, it has a key feature, which is allowing the delegation of authority of decision making to the individual nodes of this network system. And where does CanDIG fit into this landscape of federation? Well, CanDIG tries to be a highly decentralized but trusting platform to allow the federation of so CanDIG tries to be a highly decentralized but trusting federated model, which allows healthcare researchers to get access to data across the various institutions, while making sure that the data stays in place and the network is a collective of trusted federated peers. And working in this model helps us to align with the political landscape and being able to conform to the regulations and meet those political challenges. So what is CanDIG? CanDIG is the Canadian Distributed Infrastructure for Genomics, a fully distributed platform that allows a national scale, privacy maintaining analysis of locally controlled data sets. With initial sites in Toronto, Montreal, and Vancouver, participating members include some of the top generators of genomics data and in Canada. CanDIG serves as a foundational platform for all of these institutions, collaborative ventures with long-term goal of extending data sharing to a wider base of researchers across Canada. And with CanDIG, we're hoping that health researchers across Canada will be able to find and request access a much wider range of data than they were able to before, and that researchers will be able to combine cohorts and conduct analyses across sites and institutions, as well as make it possible for new projects to to join this federated group of peers and make it and ease the facilitation of new projects joining in a way that can make it easy, private, secure, and useful. While in this process of developing CanDIG, we have put a lot of work into defining the roles and responsibilities inside of our federation, defining the roles for platform, sites, and data projects, which members are responsible for data quality, for operational security and interoperability, and implementing needed features. And all of this work has been very beneficial for us to be able to work in the international stage and work towards federating abroad, as with projects like Sinica. But the GA4GH has always played a pivotal role in the formation of CanDIG and the work that we do in order to build this distributed genomics platform. And for instance, one of the key decisions and driving forces behind CanDIG is from a technical perspective, is that we build this platform to be API driven, and based on that, we needed to have a sense of which standards or API standards we could implement in order to make that happen. And the initial prototypes with the GA4GH Genomics API was very successful, and using that helped us to really refine this model of API-driven development and API-based platform to be able to create things like our distributed authentication and authorization approaches. But other than the technical perspective, we have also been able to rely on the GA4GH in our regulatory and ethics framework that we created. The framework for responsible sharing of genomics and health-related data helped structure the early discussions with our national partners and create a legal framework as well as a technical framework for us to be able to tackle this problem of data sharing within Canada. So after 2 years of working closely with real-world national cancer genomics projects such as TF4CN and Profile, we have learned from our initial prototype phase, both technically and also on project governance and policy, particularly clarity of roles between data custodians, sites, and software development teams and the platform itself. And from that, we have gone this process of re-architecting the CanDIG V1 platform to V2, which will allow us to be more scalable, more flexible, and bring in additional genomics and omics data types. And from the development perspective, it will make it easier for us to integrate new GA4GH services and standards to increase the catalog of services that we can provide to health researchers. And this talk today will go further into the experience that I have had implementing and using these GA4GH services. APIs. So as I mentioned earlier, one of the core platform design challenges one of the core platform design goals that we wanted to have within CanDIG is that all data access must be through APIs. And why this is twofold. First, using APIs provides a single interface for to be able to control the flow of data between sites, and having APIs gives us better access to be able to filter and control this data while providing fine-grained authorization decisions from the data stewards. And the GA4GH APIs were used as part of this platform design wherever possible, in order to standardize the methods for data access and retrieval. And furthermore, using these GA4GH APIs gave us the benefit of being able to abstract away the underlying data store, and moreover, just the systems that these APIs had to interact with, providing the layer, a standard layer in which other downstream applications or clients could communicate to them without having to modify the connectivity between those individual systems. And so in this V2 process, we've been using the GA4GH APIs very heavily in many places, starting from authorization to workflow execution, as well as the genomic data imaging, and are building a data catalog. And we will look at that in depth as we go forward. So starting with authentication and authorization, in order to be able to share data between different institutions, we needed to make sure that there exists a standard method in which we could verify the authenticity of researchers, as well as respect the authorization and access control for the data being requested from the perspective of the local data site in which that request was being made to, and the work that some of my colleagues at CanDIG have done have really made great strides in the adoption of the GA4GH Duri visa model for authorization, and using OPA in order to be able to facilitate these authorization requests. And now from the actual genomic streaming and genomics data access side, we were looking into using htsget, and we've initially used existing implementations from the GA4GH community, primarily HTS Nexus and Challe htsget, but during that development and implementation process, they didn't exactly fit with all of the requirements that we had in our development. So we opted to build our own implementation, which we have made available as an open source project in GitHub, and contributed back to the GA4GH community as well. And some of the reasons why we opted to build our own htsget implementation was the modularity that we could have in the back end of these from the htsget genomic streaming protocol to the data catalog and file storage, or the underlying data sources themselves. With our implementation, we were very successful in being able to modularize the backends and the htsget protocol and application can speak to, and as we increase the number of CanDIG sites and provide access to even more and more data repositories, we want to make sure that we have a modular approach to be able to connect those different data backends into and make them interoperate with our with the genomic streaming protocol that we have used in htsget. And another way in which we've used the GA4GH cloud APIs is with respect to workflow execution. In CanDIG, we've adopted this model of computation on local data sets. So that requires us to bring the algorithms or the workflows to the data and without having to copy and move them from the local sites. And workflow execution service is a great example of a cloud API which allows us to be able to abstract the pattern of workflow execution and decouple that from the actual systems in which they have to execute on, whether the system, the site supports containerized workflows or HPC, traditional HPCs. Using the WES API will allow us to have a consistent way across various sites to be able to bring the computation to the data, and also being able to use workflow execution and have that as a standard API further allows us to decouple it away from the data retrieval APIs, which can be done via DRS and htsget.
Checking in, Shaikh, if you're getting close to end.
Yes, I will then summarize very quickly here. I will skip over some of the work that we've done in integrating new services and just say that through this process, we have learned a lot and we are re-architecting and everything that as we are re-architecting and we're preparing for large national projects, we are always looking to increase our team, and we are, and in conclusion, we're hoping to incorporate new variant representation to our variant store, as well as being able to expand our efforts in the use of Duo and Phenopackets, and moreover to broaden the genomics data sets and connect them with the clinical and phenotypic data. Thank you.
Terrific. It was really nice to see all the work and the use of Global Alliance standards in CanDIG. So with that, we're going to move to the next speaker, Benilton Carvalho, who is an assistant professor of statistics and advisor to the Dean of Research at the University of Campinas in Brazil. He's also a steering committee member of the Brazilian Initiative on Precision Medicine, which he'll talk about today, and has been developing statistical methods and tools for genomic data analysis. So Benilton, take it away.
Thank you, Heidi. Hope it is fine. I hope that actually I'm finding all of you guys in good shape, in good health. Let me just make sure that I can get to the slide. Just one quick second. There we go. So you guys learned yesterday about the importance of actually having a better understanding of diversity in genomic data. What I want to tell you guys is that this is actually something that plays an important role here in Brazil. We are based in São Paulo State at University of Campinas. And what happens is that BIPMed, the Brazilian Initiative on Precision Medicine, is actually formed by a team of different professionals that are not working exclusively for the initiative. So we have medical doctors who are actually making attendances at the university hospital. And then we have to teach, we have to do a lot of administrative work as well. So what I'm gonna show you guys is actually how we have been dealing during these past few years. It's been quite an amazing ride, to tell you the truth. And what we have got so far, I have to remind you that I need to thank these 4 people here, Dr. Isia, Cristiani, Wellington, and Rodrigo, because without them, I would not have any of the results here to share with you. So this is going to be a presentation where I'm gonna show you some of the findings that we actually got in this past year through the use of strategies that we have been learning along with the GA4GH. Just to remind you guys, BIPMed is a coalition of 5 research centers in São Paulo state. And among these centers, we have specialties of different kinds. We have neuroscience, we have computational science, cell therapy, inflammatory disease, and obesity research centers. Sorry, this does not seem right. Hold on just a little bit. I need to zoom out just to make sure that there we go. I can see the whole slide right now. We also got, at the very beginning, what we had was a genomic database that was shared publicly in terms of pooled data, and it had information about subjects that were not patients. They were what we are calling reference subjects from the Brazilian population, and these databases were distributed were actually obtained through 2 different technologies. We had a bunch of whole exome sequencing and also array data. They were public shared through our system. And back in 2015, we started with the LOVD database using Beacon, and 16, we went to the Genomics API. We had a few packages delivered through, produced and delivered through the bioconductor framework. And we also had a Beacon client that was derived using R. Later on 18, what we had was moving towards a cloud environment, improving our web applications, and we had actually a specific request that we were getting clinicians to use the data that we were distributing, and they were finding it slightly more complicated for them to get a better understanding. So what we did, we provided with a new tool for us called Brave, the BIPMed Variant Explorer. And in 19, we started looking towards these newer strategies where we could actually do authentication of users and give different access, different levels of access to the data. So our databases have been initially distributed through these LOVD instances. They are databases that are actually getting data from different phenotypes. They not only have data on reference subjects, but also on specific phenotypes. And one tiny detail actually that I would like to share is that most of the traffic that we are getting on those databases are actually coming through the United States and China, and Brazil is appearing only on the 3rd place on that particular ranking. Now, thinking about the Variant Explorer, the reason that we started looking at something like that is because most of the time we would be within a programming language and wanted to query data to do, for example, filtering on variants. I wanted to see, for example, all the variants that were appearing at most 5% of that particular data set. So we implemented that, and it does support multiple data sets and different versions. Now, the thing that is actually making a big difference for us is the fact that this tool is actually transparent to the environments that we are using. So it really doesn't matter if we are, for example, working on a HPC cluster or if we are on the cloud, we can always have access to this particular tool. Now, it does have a specific API that allows us to do different queries to the data that we have stored. And it allows us to, for example, not need anymore the VCF files once the data are imported. These informations are actually required by us to be able to provide reports and do the data analytics on what we have got. So essentially, it has a web application. Let me see if I can get the, there, hold on, pointer. So it does have a component that is a web application where people could simply browse through it. And we are working on strategies to do authentication in order to be able to get to individualized data that might be needed during specific points on data analysis. And it does allow you to bring data from Python and R if that's the case that you're running into. Now, this is the visual interface that the browser has. So different types of queries can be placed there, and we could get the results, and we would have some statistics on clinical significance and also on coverage that we have for those particular samples. And later on, what we started looking at is actually the need of a better strategy to do genotype calling on our samples using GA4GH implemented best practice. Now, what we decided to do was to write actually some piece of code that is based on WDL, on WDL and Cromwell, that it does make use of the GATK4 infrastructure, and it allows us to speed up really by a lot the timing that we would get for genotyping per sample on legacy equipment that we have in-house. Now, one of the things that we have been using a lot and we need to improve our use on that is actually on the CRAM files. This has been very useful for us because it does show a lot of compression of the data. And it allows us also to have this fully reproducible and platform independent environment where I can start adding my samples and get the calls either on local machines, on clusters, or on the cloud. Now, that said, we are still on the Beacon network. We are still using the Beacon despite the fact that we have not upgraded it to the next version yet. So we probably will start doing that sometime early next year. But now, let me just go ahead and focus on one tiny thing and keep you guys on time. We have these two reference data sets. We have the whole exome data, and we have the array data on the subjects that were sampled from the Campinas region. Now, here's one information on these subjects. We have the distribution in terms of the region where those reference individuals came from, despite the fact they are now found in Campinas. Most of them are from the southeast area, and some of them were actually unknown, but we do have tags for those that were coming from the south, north, and northeast. Now, back to the subject yesterday on the importance of of knowing and understanding better the diversity on genomic data, what I would like to remind you guys is that our findings during this year that should appear soon on genomic medicine is that if you guys get markers, for example, that are called common markers, let's say they have frequencies greater than 5% on European populations, these markers, some of them, actually about 7,500 of them, are found to be rare in the data set that we have in hand on Brazilian subjects. The other way around also happens. So markers that are called rare on European populations, about 75,000 of them were actually seen as common here on subjects in Brazil. And the same behavior would happen with African populations and American populations, mixed populations in general. So what we have here, for example, for African populations is that the common markers for those set of subjects, we would have about 65,000 of these markers being called rare in subjects that we have here in Campinas. So what that brings us into mind is the fact that if we are making clinical decisions on these subjects, we actually need once more to be very aware of how those markers that our decision were based on behave on the particular population of interest, okay? Now, just to remind you guys of a famous PCA plot, you guys have here the PCAs for exome data and SNP data. I will just focus on the SNP data because it's pretty much reproducible everywhere. We have the corner there with Europeans, African at the left-hand side, Asia at the bottom right, and then mixed American populations here in green. Now, what happens is that the Brazilians are the black dots here. They scattered all over. So there is pretty much a gradient from Europe to Africa in terms of signatures of Brazilian subjects. I want you guys to pay attention. If you guys have a better zoom than what I do, behind the orange dots there, there are actually two black dots. So those are Brazilian subjects that if we were actually to look at their genome to make clinical decisions, we would actually to be able to understand a lot about the Asian genomics in terms of genomic medicine. The same thing happens here on the left-hand side where we have subjects that are getting really close, and I know that there are some overlaps. I think there are about 10 to 15 subjects that are actually underneath the red dots over there. So that's the same decision. If we were to make clinical decisions based on personalized medicine for those subjects, we would need to be very aware of the African genomic information that is used in precision medicine. So this is the first time that this was observed here in Brazil. And it's actually very important for us to take that into account in this time where we are trying to, or not only trying, we are making it happen very slowly, I know, but we are implementing the personalized medicine infrastructure in the country. Now, one other thing, and I'll save you guys 5 minutes, I'll just skip right to the end, is looking at the ancestry that we have in Brazilian subjects. And again, I'm showing these because all of these was only possible because we were using the strategies that we've been learning actually from the Global Alliance for Genomics and Health. So what we would have in this particular case is that the Brazilian population is actually an admixture of Europeans, Africans, and Native Americans. So in order for us to study a little bit better the local ancestry that Brazilian subjects show, we actually needed also to study a lot of history, right? But again, we got data on this. We got SNP arrays on subjects, on 200 subjects. These subjects were originated from different places in the country. And what we could observe really is that the history books tell us a lot already about the genetics. It tells us that the majority here of the influence that we have on the Brazilian genome, if I may force the use of this expression, is actually European, with a great contribution from Africa, and then some other contributions from the admixed Americans. So this is pretty much what we knew already, but then when we start zooming in on what happens on each chromosome, what we observed was that on chromosome 8, there is a decrease of the European ancestry contribution to the Brazilian genome. And instead, we have Native American ancestry actually peaking on that very same region. So there is this 1 point of deviation on chromosome 8. And we looked at it a little bit closer, looking at the specific genotypes. And what we were able to observe is that there was an inversion polymorphism for some of the subjects. So in red are the inverted ones, and in blue are the non-inverted ones. Anyways, once we started looking at this, we could actually start thinking about the reason of the deviations that we were observing across these subjects. I'll just go straight here and show that to you guys that what we saw in particular is that these deviation on chromosome 8 showed an increased Native American ancestry, probably due to 2 facts. And then these inversion events and recent selection were possibly the reason for these deviations. We also are making a highlight here that this is extremely important for population genomics, in particular due to the fact that events like this are actual confounders for the ancestry inferences that we might need to do in terms of precision medicine. So I'd like to thank all of you guys for having the time to hear me. And that's it. Thank you.
Terrific. It was great to hear about the data informing ancestry in the Brazilian population. We're going to move to our last speaker, Augusto Rendon, who is Principal Research Associate at the University of Cambridge and also Director of Bioinformatics at Genomics England, where he leads a team of over 50 to build and maintain pipelines for genomic data analysis in the UK 100,000 Genomes project, and the Future Genomic Medicine Service, the UK National Health Service. So as I mentioned, Augusto was taken ill, but he is temporarily, but he's nicely allowed us to use his pre-recorded talk from the GIF meeting. So we're gonna, I think Justina is gonna play that now, and then we'll follow that up with questions.
Justina, we're not hearing sound.
You might have to turn up, each of us might have to turn up our volumes. It's very quiet. I still can't hear anything.
I suppose we could go take questions for Shaikh and Benilton if we can't get this going.
Sure. Let's move to that while we're waiting to see if we can get the talk going. So starting out with questions on Slido. This one's to CanDIG. How do you manage billing, if any, i.e. the data provider is bearing the cost of the analysis requested by the external user?
In terms of CanDIG, I don't have the complete picture of how billing and costing will work in the CanDIG model. However, I can say that a lot of the in general, the funding for infrastructure in terms of hardware and software development is usually provided federally and provincially with institutional contributions. We are exploring some cost sharing models for sequencing projects and especially for the long-term storage and the maintenance costs of that data. And as for workflow execution, we'll have to think about how to actually be able to allocate those costs in terms of the number of computational cycles that are required to run those types of workflows.
Right, and so does the model where sort of like Google Buckets where you can say requester pays, does that not work here because of it being a federated analysis as opposed to download sort of environment?
Yeah, in essence, because we move the computation to the data, the requester is not necessarily the person on the hook for the cost of that computation or that request. So then there has to be the understanding of how long the workflow took to execute, how much resources it took, and how do we define the costs of those resources that were used as part of those of that request and that workflow. I can say though that we are building extensive monitoring and logging infrastructure as part of this CanDIG platform that will help us to track data usage without looking into the data itself, of course, but looking at the usage of data in respect to those computational resources from performance metric standpoint. But that performance metric data could very well play a part of that cost analysis and billing that would need to be done as part of that cost recovery.
Great. And a similar question, who funds CanDIG as a whole and who funds each province level participant? Is the participation voluntary?
Participation is voluntary. Usually there is a set of agreements that are signed between participating institutions. And in terms of funding, there are various forms of funding, like I said, whether with respect to the infrastructure that is being used or from the data sets that are being brought into the CanDIG platform. Those have usually been funded through external granting bodies. But in essence, we are still operating in the model of each institution providing what they can in order to advance to understand what works best for everyone in terms of making sure that everybody is contributing equally and receiving those benefits equally. And overall, as the CanDIG platform grows, we hope that external entities, hopefully from the federal government, will be able to provide the resources and funding in order to make genomic and data sharing in general accessible and available for all institutions who want to join the CanDIG network.
Great. And then another question for you. There were 5 other parallel national Canadian platforms in flight at the start of 2020. How many provinces, territories have fully committed to CanDIG?
That's a good question. I unfortunately don't know of all of the other parallel national platforms, so I can't say how, in terms of our similarities or anything, or differences between them, but in terms of the provinces and territories, currently we have institutions with that are based out of the province of Ontario, as well as Quebec and BC. And we always hope to increase that provincial membership to all provinces and territories of Canada.
Great.
Heidi, I'll just add to that. Since I spend a ton of my non-GA4GH time tracking and working with Canadian platforms,
yeah,
there are at least 5, maybe more. They've all got different start points, different funding mechanisms. I think the big opportunity for CanDIG is as a main host of the Terry Fox Marathon of Hope, which is going to be our coast-to-coast, eventually, cancer project. I think that will take it away from some specific smaller research projects. But each one of them have actually got a stance, and I think one of the roles that Lindsay and I play is how do you make sure that those 5 platforms are interoperable, and of course, Global Alliance, we pitch Global Alliance standards so that at least they can compete on the things that are not about interoperability. They can compete on their implementations.
Yeah. Shaikh, is it possible in the system you've built to have participants provide consent for each analysis performed on their data? And if so, how is consent recorded?
In regards to consent, we are in the process of building a dynamic consent platform, which one of my collaborators and project member, Kat, has presented at the poster presentation. So if you're interested in learning more about the dynamic consent work that we're doing, I highly encourage you to check out their poster on DICONS. But in order to provide the dynamic consent, we know that we will have to use existing standards in order to provide project level consent to data access. Specifically, the consent will have to be tied to the institutional requirements and the data of the institution and where it resides. And one way we are exploring and how to do that is using GA4GH standards like Duo.
Great. And are the same workflows repeatedly executed if different users run them?
So in regards to workflows, they are executed based on in a number of different ways. One can be for interactive analysis. If a researcher is trying to run a particular workflow and they know in which data set they want to run it against, they can make a request to run that workflow on that particular site. The workflows themselves can be federated across the different sites within the CanDIG network. But again, as with the design of CanDIG v2, we have made that decoupling of running workflows and executing workflows we've decoupled the workflow execution from the data access requests. So there are scenarios in which workflows can be executed repeatedly or across all sites simultaneously, but based on the data access level or the type of data that they're requesting, that workflow may not actually be executed because the requesting data is not available for that particular researcher or credential that's been used to execute that workflow.
Got it. And this one is for Benilton. Are there whole genome sequences available for the Brazilian data as well as exome?
For this time, for right now, what you can find is actually whole exome. We are working on the whole genome for 2021.
Mm-hmm. Great. And then Shaikh, thank you for the great presentation. How is consent of participants being managed? Are participants sufficiently informed for all uses of their data?
Right. So as I alluded to earlier, the consents are being consent to participant and data from participant is being managed right now through our DICONS platform, and I encourage anybody who's more interested in learning about the consent work that we're doing to look at the presentation from Kat and essentially right now we are still in that process of making sure that we model the same methods of consent and excuse me, where we maintain the same model of consent that's currently used in the clinical setting. So and that's where the DICON system works.
Right. Okay. And then to Benilton, this is a tremendously unique genetic variation, or there is tremendous unique genetic variation in Brazil. Are you layering any additional protections, use, exploitation for indigenous genetic sets?
All right, John, this is a very interesting question, and indigenous data here is extremely complicated for us to get. It does require a bunch of authorizations from the federal government. And honestly, this is not something that we actually have at this particular moment. Although it's always of interest, but we do have a lot of red tape to go through in order to get access to it.
Got it. And actually a related question from myself. You mentioned putting the BIPMed data on LOVD. Is that in the form of whole exome data sets, like individual level data, or is that more aggregate allele frequencies based on a cohort of exomes?
At this point, we are talking about aggregated data.
Mm-hmm. Got it. And then, you know, presumably associated with those exomes are phenotypes, which I imagine may not have consent to put the phenotypic data associated with each of those variants and aggregate data sets. But have you thought about ways to enable controlled access and query of that data, set up any, you know, standards using the search API from Beacon or anything to think about a controlled access use of that data?
Heidi, this is definitely what we are looking at right now. This is something that we want to get actually working in the first semester, 2021. And that's something that has been in the works, really. The joke that I had with Angela earlier was, can I just say that we are going to delete everything and just start from scratch? Because I have seen so many great things being shown only today. And I just want to rebuild everything now using the whole ecosystem that I saw.
Great. Next question. Brazil is very state-based country. Is BIPMed getting broad support from outside Sao Paulo?
Not really. Our support is coming from the state research agency. We are actually getting not if you're referring to financial support, no, but we are actually getting a lot of feedback from people outside the state. So now there are initiatives that are being hosted actually in other centers, and in particular in other Latin American countries. And these guys are now in touch on a constant base, trying to understand how is it that we do proper genomic data sharing in a responsible way. But in terms of financial support, no. But that's something that we would love to get. If we were able to get this particular momentum going, it would be perfect.
Mm-hmm. Got it. And another question for myself, really to both of you, as you think about the implementation of GA4GH standards. I'm curious, you can think about this in two ways. One, we all hopefully understand the benefits of interoperability and applying consistent standards to allow us to share data, enable federated analyses, all of the things that we all see coming from this. However, sometimes it requires extra work to go use someone else's standards in your own system versus just going forward in an independent, siloed way. And we all have to just think about the long-term benefits of the standards. But I'm curious whether we're at a stage where you're actually, especially when you're starting the project from ground up after standards have been built, are the standards actually enabling you to work more quickly because you don't have to develop the APIs yourself and make a lot of decisions yourself. They've in essence been made by a collective community. And so I wonder if you could comment on, how Global Alliance standards may have eased your work as well as making it interoperable versus, not yet there.
Sure, I can start off with that. So in terms of ease of work and just making the interoperability possible for CanDIG, having these GA4GH standards available allows us to have that reach of interoperability within Canada, but also following those same GA4GH standards gives us a path to be interoperable with projects at the international scale as well. Moreover, as we see the international community growing and enthusiastically adopting these GA4GH standards, more and more implementations of those standards are emerging in the open source community as fully created standalone projects. And from that, we're able to adopt those open source projects within CanDIG and feed back into that ecosystem and that development process. And so we are able to just continue to support and grow with the GA4GH community as it has been growing.
That's great to hear. And Benilton, do you have any comment about that?
Yeah. Actually, in my case, I'm just a guy who is curious about computer science. So implementation is not my strong bit at all. I'm a statistician by training, and by having the opportunity of having data sets, let me put this way, talking to each other on a federated manner, it allows me as a professional to be able to pull power from different sources. In particular, in these cases where I'm working more constantly, there are complex phenotypes where we have like these tiny contributions of sites that are completely dependent on sample size. But now, if I'm able to combine these different sets, right, with different, actually, if I'm able to pull together all this statistical power that is hidden on different data sets that are produced around the world, I'll be able to do my job properly. And if we were not to have something like this, I cannot see it happening at all.
Yeah, well, that's great. Great to hear that we're enabling. And I will just relay a comment that was made in the chat from Guillaume Bourque, who comments, the easiest part is not to have to justify our choices, which usually takes the most time. We can simply say we're using GA4GH. So well said. I agree with that. And another question that came in from Heidi Sophia, what other Latin American countries are involved in the growing network?
Yeah, we have great contributions with Mexico and Chile. On the other hand, there is also another, it's a GCRF project that I'm a co-PI with, and it's the Cabana project, where we are bringing together at least 9 Latin American partners towards these bioinformatics solutions to build local capacity.
Great. I think so Justina does have Augusto's talk. It's about a 10-minute talk, so we have plenty of time for it, and before we need to wrap up. So I'm gonna actually turn it over now to Justina so we can play Augusto's talk.
Yeah, good morning, afternoon, evening, middle of the night, early morning. So thanks a lot for the opportunity to speak. I will give a quick overview, when I manage to get this working, about what we are doing in Genomics England, particularly in light of GA4GH standards. And I will do so not so much from the perspective of as a researcher, I would like us to federate and share data, but rather from like an engineering and a products perspective of why these standards are helpful. And for those of you who were in the previous forum earlier this year, it's not dissimilar to that. So in Genomics England, we have two main aims. We are there to help evolve the genomic healthcare in England and wider in the UK. And we do that primarily by serving the NHS as a diagnostics facility where we have requests for a analysis based on whole genome sequencing for patients with rare diseases and cancer. And we provide the full stack all the way back to results going to the labs. And then as we are accumulating those results subject to consent, those participants' data, then we use that to fuel research by taking those data and presenting them to users, academic and from pharmaceutical companies, startups and so on, to be able to make discoveries that in turn flow back into the improving health. And indeed, we have a number of examples, hundreds, where research has led to new diagnosis in the NHS. So this very now common paradigm of its infinity loop is precisely how we see our role in the ecosystem. Good. So if I focus on that first healthcare loop, briefly, what we do is we receive a test order from the NHS. We sequence data through a human technology that comes to us, and we do alignment and variant calling. We do so by orchestrating a number of pipelines and storing some data. Once we have those alignments and variant calls, we do the standard that scholars have presented before around variant interpretation, annotation, prioritization, those sorts of things. And we do so by applying a number of what we call interpretation services. And for example, this pharmacogenetic analysis that Mark was just speaking about is one of those additional findings, but there's also ways of prioritizing and classifying variants in rare diseases and cancer. And finally, we make that data available for the labs on the right-hand side. We call them genomic laboratory hubs in the new genomic medicine services in England. And those labs have access to those data, and they do various things. As you'll be familiar, they would browse those genomes, perhaps through a standard genome browser like IGV. I'll show you an example in a second. Or they will review the variants and the clinical information and the scoring of those variants to be able to diagnose those cases. And finally, those data are deposited in variant knowledge bases. So as we're doing all of this, there's a number of challenges, and we have started to implement more and more GA4GH standards to solve those challenges. For a while now, we've been using CRAM as our data storage, and that essentially saves money, but also the tool chains now to run those pipelines and so on are by and large fully compatible with CRAM. So there's no need to inflate and deflate from CRAM to RAM and so on. So that's been very helpful. The one where we are perhaps most mature of all the other standards is htsget, and I'll just in a second present a slide on that. So I'll leave that for a second. But it is our primary way of communicating genomic data to our network of laboratories. One of the big problems we've had in the way we run pipelines is that there are teams that are doing R&D trying to establish new pipelines for cancer, for rare diseases, for cancers that have high contamination in the normal from the tumor, those sorts of things. But the process of taking those pipelines and then bringing them to a production system has been quite complicated and tedious. So we have started as proof of concept, as I say here, seeing how we can use just the standard workflow execution services from GA4GH standards and adopting tools such as Chrome that was mentioned earlier to match both what would be the ISO created to clinical standards pipelines, but be able to flow in the R&D quicker into it. The same case here where we, this is slightly simpler services, so we're using the task execution service and it helps in the same ways. Very similar story here. If we move into the data space, we have the data repository services. One of the big problems we have is we have data both in our data centers, but also on the cloud. And it means that mounting directories and knowing what file is where and what do I have to, how do I access it, has become a serious hindrance. And therefore adopting this standard is very helpful so that we can have a single way of identifying a file in the world, regardless of whether it sits on-prem or on the cloud or in someone else's cloud. If we move forward now into the future, then we have this very knowledge bases, and we heard yesterday from the ClinVar folk that that they are starting to open APIs for submission from their knowledge bases. So this is an area where we are participating quite actively, both through GA4GH, but also from an implementation perspective, seeing how we can use those standards to represent those represent variants better. And finally, we strongly believe that we need to share that information across the world. And so things like Beacon2, where it's not just do you have this variant, but can have a lot more information about that variant, something that we're involved right now, certainly in the scoping and following closely, but we haven't started to consider the implementation. So this is, I hope, gives you an idea of us, how we're using those various GA4GH standards, not for the great ideas of federation and data sharing, but really just to solve some of the engineering problems that we have at present. Let me spend just a second on htsget in particular. So for us, Genomics England holds all the CRAMs and the VCFs for the genomic medicine service when it comes to whole genome sequence. But the labs, as you'll be familiar, quite often need to look at those variants and look at the read level support to ensure that they are comfortable reporting that variant eventually to a patient. And so the previous ways we had to disseminate that data were quite clunky. And once htsget came across as a standard, we decided to go ahead and use that, and we created an implementation that is relatively well performing. It's open source, and you're more than welcome to give it a try if you're desperately looking for an implementation for htsget for your own solutions. So let me give you kind of an idea of how that is. Here is just showing you the, if you're familiar, just the API call, htsget, where you can give what formats and the positions, and it will, with authorization and authentication, it will return the data. And you can use that in a number of ways. Here is simply calling it from some tools, where you can provide nowadays, because the toolchain supports htsget, you can just provide the htsget URL, and you can see here the resulting BAM, or the CRAM, I mean, and you're exporting that as a BAM. But you can also load it into a browser. And so here is the rather clunky browser that we have. Is there based on IGV, but you can also select files that you want in there. And it's just showing you from a lab perspective, being able to get data centrally across the country. That's been quite a lot of success, and it really has solved a problem that before we had a number of alternative standards. The second one is more from the research loop. So in this case, we have a research environment where our data, and by our data, I really mean the data from the participants, and we are custodian of that, but just shorthand for it, is read-only, so you cannot download the data from the research environment. That is the commitment we've made to our participants so that their data is kept safe. And so that's a pretty high barrier for people to just come and use it. People are normally, users are normally expecting the institutional HPCs or the desktops. You download the data, you have all the tools, you're familiar with those environments. And so the question is, how do I bring my data? In this particular case, how do I link a repository, or how do I bring my own workflows? And that's that task repository service. And how do I execute those workflows? And that's workflow execution service and task execution service, particularly without the need to go through a steep learning curve to learn yet another, here's GEL's new environment, or here's UK Biobank's environment, so on and so forth. I don't want to spend a lot of time trying to learn. I just want to use the data. And so that means that for us, we can have more users and tell the government that we have lots of users writing lots of great papers, and please keep funding us. So just from like a simple perspective of getting the data used, that's fantastic. But the alternative is you have to learn another workflow execution service. Perhaps you have to become either familiar with Nextflow or WDL. Not everyone is familiar with those. You have to learn how to use GitHub very well so you can bring code in. Same with S3 to bring data and so on. So this service just provide a single standardized way for the community to just go ahead and use them. And yeah, I made the point here that actually it's a federation in the case that to bring all these data sets, to come and use it together, rather than a lofty goal, is really just a way of achieving our mission of getting the data used. So one of the kind of most exciting things that I think I find in this space is the very simple what's happening in the cloud workstream in GA4GH. And here's just an example. This is Docker store where various the workflows are maintained. This particular one is WDL. And the point is that if I have the right credentials, and there's another workstream on that, then I can go ahead and launch this tool in any of those ones. And what we are working towards is to add something here that is a GEL trusted research environment. But it means that as a user, I can have easy access to the tools. I can bring in more tools, and I can deploy it across a large federation of repositories. And we'll hear, I'm sure, much more about this for those of you who are going to be at the GA4GH meeting next week. Great. So in summary, I would just like to emphasize the point that GA4GH standards are not just there for genomic research, but it's also there for clinical standards. And some of them are ready for production systems. htsget works really well, WES and TESS, they're used by many people, as well as the authorization, authentication, and passports are becoming quite robust. One of the big problems, though, is that the implementation is challenging, and so implementing those such that they're performant at the scales that we need them to be is difficult. And so some of the cloud vendors are doing some of that work, but not everyone is on the cloud. So I encourage us to share them, because if we don't share those standards, we're just not big enough as an ecosystem to have many different implementations, people coming to them. We need to share them and not reproduce too much work. And then the last one is just a call. There are many evolving standards, and we've heard about some yesterday, Phenopackets and pedigrees, but there's also Beacon 2, ways of thinking about interpretation and evidence reporting. And they're only going to be right and therefore useful if we are involved in participating, at least providing feedback, but ideally in those communications. So it's a call then to be part of that, so that we can all one day be able to interpret in better ways for our patients. Thank you very much.
Great. Well, as I mentioned, we don't have Augusta with us in person, but as Angela posted into the chat, you can still post your questions. I see one already for Augusta, and we can pass them on to him. If anybody is here working with Augusta's team, you can also reply on his behalf. But with that, we are actually right on time, and so I'm going to turn it over to Peter for some closing remarks.
Thanks, Heidi. A great conversation, and congratulations. It's, I think, one of the things that's been really great to see in the last year or two is just how many implementations we're getting, and now increasingly using multiple standards. So congratulations and thank you to all. This does conclude track 2A, and we've done so on time. So again, thank you to everybody for doing that. we really started the morning with this look at the story so far, with both the video that we've seen many positive comments on, and then a Q&A with our workstream leads. The video builds or is an expansion of the animated video you saw yesterday. And for me, it's best seen in context with the kind of things we saw in the connections demo on the breakout, or that Heidi led us through yesterday. So if you've not seen all of that, the complete suite of the story so far, then I would encourage you to get the different pieces. And Andrew, I'm sure we can find a way to put those places in a simple, easy to put together way. Breakouts, I could only attend one of them. It's a challenge of breakouts when you're not able to just move from one to the next easily. But it was a very well attended and a very positive breakout on Connections 2020. And I hope the others were as good, and that people did manage to get to see it. And I see that Andrew is already chatting where the videos are available. In terms of the work we just saw, apologies for that little glitch, and thank you, Justina, for fixing it, and Heidi's flexibility in moderating through it. 3 great examples of real-world implementations. We want to see more. We want to find ways of helping the community learn. We see it in GIF already. So the Global Health Implementation Forum is really an opportunity for small, emerging, new national initiatives to learn from the work that folks like Australia, Genomic Health Alliance, and the work Augusto and others have done at GEL, and is now being modeled in some other large-scale ones, but huge learning that other people will not have to go through. And so I think the more we can get these real-world implementations, and particularly the learnings from them, the better. So this was track 2A. For those of you whose time zone or energy level will permit, I would encourage you all to come back for track 2B. There will be parts of it, the first parts of it will be a repeat, so you could miss the first couple of sessions, but the real world implementations will be very different. And in fact, we'll have a different panel doing the Q&A on the work stream. So even that may be the same title, but we'll have different presenters. So for me, the more of this plenary, this virtual plenary you can attend, the more the experience integrates itself together. And I would encourage as many of you who can to come back for more implementations in track 2B. And this is the agenda for this. So the story so far will be repeated, breakouts will be repeated, and then there'll be completely new implementations from Australia and Japan. I mentioned earlier this morning that one of the most fun, new, innovative pieces of Global Alliance is spatial chat. If you haven't experienced it, I would highly recommend it. And I think at least a couple of us will head there now, and you'll probably find us in the bar. So with that, I would like to thank you. Oh, Heidi's coming video, so I think she's heading there already. I'm sure Angela will be just behind her. So thank you again to all presenters, all panelists, everybody who made this session work. I know how much work is involved up front and on the day. So thank you to everybody, and see you either in spatial or hopefully at our next GA4GH session, whether it's a few hours away or a few months away. Thank you.